All right, so here we're going to discuss the pentose phosphate pathway. And so it's important to understand the importance of the pentose phosphate pathway in the generation of NADPH. It's one of the ways that your cells can do this, and it's a very important way for your red blood cells to do this. So we'll just discuss the reactions in the pentose phosphate pathway and also what happens to the carbons. So they can kind of go in a few different directions. So this is considered an anabolic pathway, and this is going to be important in generating NADPH. NADPH is going to be used by cells for different reasons. Your liver cell, for example, use NADPH when it's going to detox compounds. Your red blood cells will use NADPH to keep glutathione reduced so that it can interact with reactive oxygen species and protect the cell membrane. Another reason for pentose phosphate pathway is to generate our ribose, our 5-carbon sugars, which of course is going to be important for cells. We have to have ATP, which is going to use the 5-carbon sugars. Many cells need to divide, and they're going to need to make nucleic acids with that. And so you can also metabolize dietary pentose sugars as you're breaking down nucleic acids, so you're breaking down your purines and pyrimidines. And then we're also going to be able to feed back into glycolysis if that is necessary. All right, so just a little bit of a difference between NAD and NADP. Enzymes that function in a reductive direction are going to use NAD, NADPH, and our oxidative enzymes are those that use the NAD, NADH. Now, there are a few names for this pathway. It's going to be called the phosphogluconoside pathway or the hexose monophosphate pathway, hexose monophosphate shunt. And non-dividing cells, again, like our red blood cells, the key for this is to generate NADPH. Dividing cells, of course, will have to use those 5-carbon sugars. So pentose phosphate pathway is kind of, you can think of it as a bypass of glycolysis. So in glycolysis, of course, we're going to take glucose 6-phosphate and then generate pyruvate and generate ATP in this process. Now, the pentose phosphate pathway is going to use glucose 6-phosphate and generate our 5-carbon ribulose 5-phosphate. And then ribulose 5-phosphate can go to make nucleotides, or it can feed back into, through non-oxidative pathway, back into glycolysis. So there are two general phases. There's an oxidative phase and a non-oxidative phase. And so during the oxidative phase is where you're going to generate NADPH, and we're going to generate 2 molecules of NADPH in this process. You're going to have glucose 6-phosphate enter, and you're going to have a glucose 6-phosphate dehydrogenase, which is a key enzyme. This is a very important enzyme. The amount of glucose 6-phosphate dehydrogenase within a red blood cell will determine the lifespan of the actual red blood cell. So the more it has, the longer the red blood cell will likely survive. And then you're going to have your NADPH generated through this dehydrogenase. And then you're also going to have a second dehydrogenase in which it's going to generate the second molecule of NADPH. So 2 moles of NADPH are going to be made per mole of glucose that enters this process. Now, we're going to use magnesium in this process, and we're going to generate our ribulose 5-phosphate eventually, and then our end products in the oxidative phase are going to be carbon dioxide. That's through that second dehydrogenase. And then we're also going to generate, of course, NADPH and our ribulose 5-phosphate, which is our 5-carbon sugar. So we enter with glucose 6-phosphate, 6 carbons, and we end up with a ribulose 5-phosphate. Now, glucose 6-phosphate dehydrogenase deficiencies also are leading to something called favism. Fava beans are going to be high in our glycosides, and this can generate reactive oxygen species, and so glutathione is very important in removing those reactive oxygen species. This is also the case, as I mentioned before, for red blood cells. Red blood cells depend on NADPH to keep glutathione reduced in order to protect against reactive oxygen species. Ribulose 5-phosphate is going to become ribose 5-phosphate, and this is just conversion through an isomerase. And then you can have your nucleotide synthesized from this precursor, or it can go back to glucose. So if you don't need to make any of your 5-carbon sugars, you can then go to the non-oxidative phase and re-enter glycolysis. The non-oxidative reactions are going to be reversible. We're going to have, in this case, we have isomerases and epimerases shown here. And fructose 6-phosphate is going to be converted to the 5-carbon sugars and back and forth. You're going to have a series of rearrangements and transfers that are going to convert that ribulose 5-phosphate to ribose 5-phosphate and xylose 5-phosphate, and then many different enzymes are going to jump in, epimerases, transketolases, transaldolases, to generate back to the glycolysis. So in this case, we have glucose 6-phosphate, and it's a 6-carbon, we know that. And then this 6-carbon is going to enter the oxidative reactions and generate a 5-carbon. And then we're going to want to get back to glycolysis, and we're going to do this through by regenerating again another 6-carbon or 3-carbon. Okay, and so the whole purpose of this non-oxidative is to get to the point where you can have 3 carbons to enter glycolysis in the form of glyceraldehyde 3-phosphate, or 6 carbons, which is fructose 6-phosphate. And so you see here that in order to get the products at the bottom to enter glycolysis, you're going to need 3 moles of glucose to come through. So 3 moles of glucose come in, you're going to make 6 NADPH, 3 carbon dioxides, and then you're going to make 3 ribulose 5-phosphate. 3 ribulose 5-phosphate is going to be 15 carbons, so it's 5 times 3 equals 15 carbons. And here you have 6 plus 6 plus 3 is 15 carbons. So 6 plus 3 equals 15 carbons. And so all these steps in between are simply just to take those 15 carbons that enter and make sure they get into the 15 carbons that can exit. And so you have a transketolase, you have a transaldolase that's going to be used. The transketolase, again, is going to be used over here, etc. And again, it's just to get those carbons back. Some cells might need to make nucleotides, and so they would use those carbons for that, of course. So there are some disorders, and these are going to be have some severe deficiencies, for example, of thiamine. So this is a component of thiamine pyrophosphate, which is a coenzyme TPP that's going to be on all our transamination reactions. And a syndrome is in alcoholics, and so alcohol interferes with the absorption of thiamine. Mutations of that transketolase is going to exacerbate this syndrome. The transketolase, and again, that pentose phosphate pathway, those enzymes are important when you have your degradation of your 5-carbon sugars as well. So mutations of transketolase will lower the affinity of the TPP about 1/10. And then this, of course, can lead to many symptoms, just simply because you can't metabolize your 5-carbon sugars as well. So when do cells go into the pentose phosphate pathway? Well, it depends on the need. So do you need to make NADPH? What is the ratio NADP, NADPH? If you have low levels of NADP, then you cannot go through the glucose 6-phosphate dehydrogenase. So that first step will be blocked. So NADP is going to activate glucose 6-phosphate dehydrogenase to generate more NADPH. Now, what is NADPH going to be used for? Again, it depends on the cell. We have our reduction of glutathione, and this is important for protecting against reactive oxygen species. And then we have our cytochrome P450, and this is going to be our detox, especially in our liver. They have a lot of P415. We're also going to use this when we make fats. So when we talk about the synthesis of fatty acids, it's going to use NADPH. Also, cholesterol synthesis is related to fatty acid synthesis. Some neurotransmitters are going to require NADPH. Nucleotide synthesis, of course, that we just talked about, and superoxide synthesis. Our glucose 6-phosphate dehydrogenase is going to be found in our phagocytic cells, and this is to generate superoxide so that they can protect against pathogens. And then, of course, NADPH is a feedback inhibitor of glucose 6-phosphate dehydrogenase. So rapidly dividing cells such as your immune cells are going to require a lot of NADPH. We're going to generate superoxide radicals that use NADPH, such as your macrophages. And as I mentioned before, erythrocytes need a large amount of NADPH in order to defend against reactive oxygen species. This is an image of your red blood cell and what red blood cells do. So red blood cells, of course, always pick up glucose. That's their only fuel. Glucose is generally going to push towards glycolysis. It can go through the pentose phosphate pathway in order to generate NADPH, which is very important because NADPH is going to ruse glutathione, and then glutathione is going to help to protect against reactive oxygen species. If you have a glucose 6-phosphate dehydrogenase deficiency, this is going to compromise this whole glutathione defense system, and it's going to lead to your cells being killed faster. So this reduction of glutathione is very important for cells like erythrocytes because they don't have any other defense mechanism. Okay. And so in general, keep in mind that the pentose phosphate pathway is going to have an oxidative phase and a non-oxidative phase. Oxidative phase is to generate NADPH. You also generate 1 mole of carbon dioxide because you're going from a 6-carbon to a 5-carbon. You get ribose 5-phosphate, which can go to make nucleotides for nucleic acid synthesis, or it can go back through the pentose phosphate non-oxidative with transketolase, which has that TPP as a cofactor. And then you're going to convert back to either a 6-carbon through fructose 6-phosphate or a 3-carbon in order to re-enter glycolysis. If you have a defect in transketolase, you're going to have some syndromes associated with that. And then, of course, NADPH is a negative inhibitor of glucose 6-phosphate dehydrogenase. So high levels of NADH, you will turn off the pentose phosphate pathway.